So our next presenter is Julian Dupuis. His talk about comparing population genomic strategies for developing diagnostic markers for a maxfly and Anastrepha ludens. Please.
Thanks, everyone. I'm going to try walking around with this. So yeah, thanks everyone for coming, and we're going to shift gears a little bit. I'm only going to talk about one species for the next 15 minutes, which is a bit of an oddity in this conference in general. But the whole kind of purpose of this talk is to think about using genomic data sets to create molecular diagnostics, in this case for Mexican fruit fly. And I work on many projects in my group along those same lines, and they all share the same concept of taking as big of a genomic data set as we can get and going through filtering and analysis to basically whittle that down into a very small number of markers that are very highly informative for a particular diagnostic question. And these questions vary from asking things like, what is it? Species identification, species delimitation, often in a phylogenetic context, and where did it come from, which is important for geographic source determination of invasive species, and often more in kind of a population genomics context. And obviously, both of these questions are important for Tephritidae. As we've heard, their taxonomy is a mess in many cases, and they're very important economic pests, or else I wouldn't be speaking in the symposium of Tephritoids of economic importance. And so both of these questions are really important. And when I think about this task of taking big genomic data sets and whittling it down to highly informative markers, I basically think about it as playing diagnostic marker limbo. The question is, how low can you go? How many markers do you actually need to answer particular diagnostic tasks? Now, if you're not familiar with limbo, there's a very synthetic, intriguing article by the researchers at WikiHow with wonderful illustrations, so just for your future reference. Now, this introduces kind of an interesting conceptual dichotomy, because genomics is all about bigger, bigger, bigger. More data, more data, more data, faster data with these high-throughput sequencing platforms. But in this task, we're taking the biggest data sets we can possibly get and whittling them down to the smallest data sets that we need for particular questions. And this involves two interesting questions at different ends of this dichotomy. You know, at one end, I already mentioned, how low can you go? How many markers do you need for a particular question? But at the other end, is bigger always better? Do you need the biggest genomic data set that you could possibly get to develop diagnostic markers for particular questions? If you take maybe a slightly older genomic approach like RAD sequencing, we're using restriction enzymes to target random spots in the genome, and usually end up with maybe a few thousand SNPs in a filtered data set. Is that just as good as sequencing the whole genome of all those organisms and ending up with 1 million SNPs in your data set? This is kind of a core question in population genetics. How many individuals do we need to sequence, and how many markers do I need to answer the question at hand? So those are the two big questions that I'm going to address in today's talk, and with the Mexican fruit fly, Anastrepha ludens, or Mexfly as I often call it. So obviously it's a Tephritidae of economic importance, native originally in southern Texas, although we've extirpated it down, and now found in Mexico and down into Central America. Although you can see from the climate suitability map, you know, Southern California, Florida, and Southern Texas are all suitable habitats for this species, and it shows up there pretty regularly. And so when that happens, quarantines are established, and IPM is engaged to basically try to deal with these pests. The other issue with, not issue with Mexfly, but other characteristic is that we have pretty effective sterile insect technique. So we can rear billions of these flies up, irradiate them, sterilize them, and wash them over areas where the incipient invasive populations are to basically squelch any reproductive capability of fertile wild flies. And there's 3 main strains that are used in Mexfly. Tephritoidea 7 and family 10 in Mexico and Guatemala are black pupil strains, so we can only release male flies. And Willisii is a bisexual strain brewed in Texas. And we can separate SIT flies from wild flies pretty effectively using the standard methods of coating the puparia in this fluorescent powder, which gets incorporated into the head capsule that can then be viewed under UV light. But oftentimes, USDA, APHIS, and PPQ have situations where they want a second opinion on whether a fly is a wild fly or an SIT fly. You know, if you're missing a head, it's going to be hard to look for a fluorescent head on a fly in a trap. And this is a pretty common question within Tephritidae of finding molecular markers to delimit wild flies from mass-reared flies. So here's the case of medfly, which has been mass-reared for decades, and they have a single SNP that will delimit very effectively all wild flies from all mass-reared colony flies. So what I'm going to ask in this talk is basically those two big conceptual questions I introduced at the beginning with regard to Mexican fruit fly, and using both ddRAD, which is a rad-seq method that like I mentioned, ends up in data sets of about a few thousand SNPs, to whole genome shotgun sequencing or whole genome resequencing, which usually ends up with hundreds of thousands or millions of SNPs in a final filtered data set. And we're basically asking three main questions. Can we develop diagnostic markers to separate wild flies from mass-reared flies? Can we separate the three mass-reared colonies? And can we separate wild populations themselves? And this is work that I started years ago, but now I've got two postdocs who have worked on it, Oksana Bernigora and Kim Vertasnik here at University of Kentucky. So we started off actually with a pretty decent foundation here in work that I did several years ago of doing basically the wild population genetics of Mexican fruit fly. So I'm not going to dwell too much on this, but we ended up sequencing, close to 400 individuals. Again, ended up with a data set of about 2,000 SNPs. And overall, we found four main genetic clusters corresponding to Western Mexico, Eastern Mexico, up into Texas, and then a couple clusters in Central America. And then we played diagnostic limbo with this data set. Taking our 2,000 SNPs, how low can we go? How many do we need to actually recreate the genome-wide population structure? And the answer in this case is about 32 SNPs. So with 32 SNPs, we can roughly recreate the same population, genome-wide population structure that we observed with 2,000 SNPs. And then we actually went and developed an assay for those 32 SNPs and sequenced intercepted specimens and could trace them back to their populations of origin. So this is where we started off here, but one thing that was missing, of course, with regard to thinking about SIT is we didn't really sample any SIT colonies. So we've started basically sequencing more individuals, and we've kind of gone a little hog wild with sterile insect technique colonies and have sequenced, probably now close to 900 specimens from mass rearing colonies. And for population geneticists out there, you might think that's kind of a really stupid thing to do. You've got an inbred bottleneck mass rearing colony. Why do you want to sequence hundreds of individuals? And I'm not going to go into all the questions we're asking, but we have more kind of mass rearing efficacy and quality questions at play. But so the biggest ddRAD data set that we have now is about 1,200 individuals, and we again end up with about 2,000 SNPs in this data set. And through either ordination-based analyses or kind of structure-based analyses, we can find similar population structure in the wild, especially in structure. We can at least find clusters corresponding to Western Mexico, Eastern Mexico, and Central America. And then we find nice clean clusters of the three mass rearing strains, Topsula 7, Family 10, and Willacy. And you can see in some analyses there's a little bit of kind of intergradation or mixing between those clusters, but overall we can generally separate them pretty effectively with a few thousand SNPs. So again, at this point we want to start playing diagnostics limbo. How low can we go? And so we can look for highly differentiated markers, and genome-wide we can do that either by thinking about FST or looking at allelic loadings from ordination-based analyses. And we see highly informative markers spread across the genome of Mexican fruit fly. And we've got a really good reference genome for this species, which is nice. And Oksana made a nice little program called SNPcheck that's publicly available on GitHub if anyone's doing this kind of analysis that basically takes these set of markers and does statistical leave one out kind of cross-validation and thousands of replications to address how efficacious each marker is at taking an unknown individual and reassigning it to its population of origin. So in the question of basically finding markers to delimit wild versus SIT flies, if we take, say, the 20 highly differentiated SNPs, and I should have mentioned those are just these random red arrows on this FST plot, and address how good those SNPs are at telling an SIT fly from a wild fly, combined they're about 99% successful. So that's a pretty high success rate of telling it's an SIT fly or it's a wild fly as the question. SNP to SNP, there's quite a bit more variation, and you can see some of these outliers, are getting kind of down more towards 50 or 60% success. But if we take, say, the top three, least variable of these SNPs, we're still at about a 95% success rate. So that's pretty darn good. We can take random combinations of these SNPs and address their efficacy. And again, anytime we get above maybe 2 or 3 or 4 markers, we're above 95% success in telling wild from SIT flies. Now that seems pretty good, but it's kind of cruddy compared to the previous example I talked about with medfly. That was a single nucleotide in the genome that can tell all mass rearing from SIT, all wild from SIT flies. But the important thing to remember is medfly has been mass reared for decades at this point. And we include, Willacy, which is established in 2008 from wild flies in Texas. So that's a more difficult diagnostic question to ask because that Willisii colony is going to be much more closely related to wild Texas flies than, say, medfly that have been mass reared for decades on end. So what about these other diagnostic questions? Can we delimit the SIT strains from each other? And again, we can pick those out in our population structure analysis. And if we take the top 20 SNPs, we can see we have about a 98% success rate. 6 SNPs gets to about 90%. And random combinations, even going up to about 10 of these highly informative markers, only get us about 90% successful reassignment. So a little bit less successful than the previous diagnostic question, but again, it's probably a more difficult one. We're trying to separate out these mass rearing colonies from each other, not just the mass rearing colonies from the wild flies. Same thing with the wild versus wild comparisons. In the original ordinations I've been showing, the first 2 discriminant axes can't even separate those wild population clusters. But looking at, say, the 4th discriminant axis, we can separate at least West Mexico from East Mexico from Central America. And again, looking at the best SNPs, we're at about an 85% success rate of reassigning unknown individuals to each of those population clusters of origin. So a little bit lower success, and interestingly, much less variable than any of the SNP comparisons when we're dealing with SIT flies, which is probably some fine details within this population genetics data set that could explain that. So the next question then is bigger always better? So what about using shotgun sequencing? So we've basically taken our big DDRAD data set and taken a subset of about 180 individuals and generated a shotgun data set from that. And filtered, we end up with about 180,000 SNPs. So about a few orders of magnitude larger of a starting data set to look for diagnostic markers. Interestingly, the population structure of these data sets are pretty similar, although this one, the ordination is kind of flipped up on its end, and I'm not really going to get into kind of digging in the details of the wild clusters here. But despite the similar population structure, when we pull informative SNPs from these 2 data sets, here we've taken kind of a subset of the 1,200 individual DDRAD data set and looked at informative SNPs, and you see they're getting maybe about 90% as the best ones. Compare that to the shotgun-based SNPs. The shotgun-based SNPs are all at least 90% successful, if not up towards 95, as an individual SNP. On average, those shotgun-based SNPs are 12% better at telling wild flies from SIT flies as compared to DDRAD SNPs. So is bigger always better? In this case, yeah, it's exactly 12% better on average. And so when you're thinking about diagnostic marker development and creating these kind of big genomic data sets, you kind of have to consider, well, what are the questions at hand? Are they difficult diagnostic questions or relatively simple ones? How much money you have, of course, and what kind of success rate you want from your diagnostic markers. If spending relatively less money on some DDRAD data, but having a slightly less efficacious diagnostic marker is fine, then that might be the right choice. And ultimately, it's all about whether you want to win or lose at this diagnostic marker limbo. And not really lose, that's a harsh word, but you get my point there. So in answering these 2 questions I laid out, is bigger always better? Yes. And in MexFly, it's 12% better to get a shotgun data set as opposed to a DDRAD data set. And how low can you go? Well, that's a pretty subjective question. You can go pretty low, but it's going to depend on your diagnostic question at hand, if it's a simple one versus a more difficult one. And so we're currently kind of pulling all of these SNPs together into kind of a one-stop shop diagnostic panel for MexFly to answer all of these diagnostic questions at once, which is something that APHIS and other regulatory agencies have a need for. So with that, I'd like to thank especially Oksana and Kim again in my lab at UK and a lot of collaborators here working on Tephritidae, and if I have time, I'd be happy to answer some questions. That's a good question. So the black pupil strains in Mexico and Guatemala, top 207 and family 10, are older mass rearing colonies, and they actually get infused more frequently than Willisii does. So I think they just have local infusions going on there. So you could probably address that more directly for those 2 black pupil strains with dedicated analysis. But it makes sense, at least with Willisii, that we see it very closely related to the wild Texas flies as compared to the other colonies from other geographic origins. So I feel like that's a half answer to your question. I haven't exactly addressed, especially the black pupil strains there, but for Willisii, you can kind of see where it's coming from, that answer.